All right. So all the testing materials are, as I told you, shown on the green sheet. So make sure you go through it. If you know all the answers to these questions, you should be okay. All right. No need to do additional um, notes or anything like that. Just make sure you get really familiar with these things. All right, so first we have 5 questions regarding the beetroot lab. Like I said, the beetroot was done fairly well, so I'm not too concerned. There are a few questions on the test, all in the multiple choice for the beetroot experiment. First question, what is the control in the experiment? The control would be the room temperature. Room temperature test tube, right? And the purpose of including control is to have a reference to compare other treatment groups with, okay, to have a baseline, to have a reference. Next one, what is the dependent and independent variable of the experiment? For the dependent, okay, yeah, so independent is the temperature. The dependent, I would say, is membrane permeability. Membrane permeability. And this can be measured in one of 2 ways. We observe it qualitatively, just by looking at the color intensity. On the other hand, even though we didn't have the instruments to actually measure the intensity, but I explained to you, you can actually use a calorimeter to measure the absorbance of green light. Right? So both of them are different ways to get the membrane permeability. And the independent variable would be the temperature. Okay, moving on, we have what variables are standardized. There are a couple of things you can say. The volume of water in the test tube, not so much the beaker. The beaker doesn't really matter. As long as the temperatures are even in the beaker, that's okay. We have the size and shape of the beetroot discs. Okay. They should also be from the same beetroot. Okay. You cannot have this from different beetroot. That would definitely affect your results. Some beet might have more color than others, right? One more independent variable would be time submerged in the beaker. Okay, so when you expose the beetroot to different temperatures, you want to keep them in for 1 minute, right? I believe that was the time in the lab. Yeah, so like 80 degrees for 1 minute, 70 degrees for 1 minute, so on and so forth. So these are standardized variable, right? Why does a frozen beetroot have similar color intensity as 80 degrees Celsius when placed in water temperature? This one I explained in class. I think if you were there for that class, you got the answer right. Freezing damages the cell membrane due to formation of ice crystals. Right, so all the ice crystals are gonna pierce the cell membrane, and it's gonna be full of holes. So when you thaw it, it's already damaged. Everything's gonna leak out. It doesn't really matter what temperature you put it in. Okay. Freezing damages the cell membrane due to formation of ice crystals. All right. What do you expect the absorbance to be if we included temperatures higher than 80 degrees? You might remember that the graph looks like this. Okay, it started not rising too sharp, and then it goes sharp, and then it kind of plateaus, right? Okay, so like in the lab, you have to label these things. This would be absorbance. If you didn't label, you will lose some marks. This is temperature. And it peaked at around 70, okay? Peak means it doesn't change substantially afterwards, okay? So a lot of you say it peaked at like 80 degrees, which is like not correct, but I give you the marks anyways. So like it peaks earlier because it doesn't change substantially afterwards anymore, right? And so this question is asking you what happened, like we 80 was the highest degree that we went to, highest temperature. Like what if we include 90 degrees or 100 degrees, right? What do you think will happen to the absorbance? Well, judging from the pattern of the graph, it's not like it's going up, right? So I would expect it to be similar absorbance as 80 degrees. Okay. Why? Because the membrane is already completely destroyed by the time you get to 70. Okay. 70 or 80. It's completely gone anyways. Like heating it more is not going to give you more color. It's completely non-existent at that point. So absorbance is going to be similar to 80 degrees. All right, that's that. Don't worry if you can't copy everything. I'll post this online afterwards. All right, next part we have the scientific method. And remember, these purple stuff you don't have to know, okay? No questions on the test for the purple stuff. Okay, list the characteristics of living things. I mean, you should be pretty familiar with this stuff already, but it's okay. We can label it. So complexity, okay, ability to maintain homeostasis, respond to stimulus or stimuli, energy requirement, reproduction, adapt and evolve. I'm missing one. What am I missing? Maintain homeostasis. I got the energy here. Oh yeah, thank you. Growth and development. I'm gonna fail my own test. Growth and development.
That can be like grows and evolve, like, is that okay word, like,
Yeah, I mean ballpark word is okay.
okay.
Remember those 7 things should be okay. Okay. It's just gonna be a multiple choice question anyways, okay, for this particular one. I'm not gonna ask you to list them. Okay, what is a Palsebal? Palsebal is a sham treatment. Okay, so sham treatment, like it's not real, right? That's basically what it means. And you would need to have a placebo to minimize bias. By the way, on the test, if I like, for the quiz, there are some questions where I give you a bunch of lines to answer, right? Okay. Then use full sentences, please. Okay. Don't use point forms or just, create your own chart or whatever. If I give you lines, full sentences. Minimize bias and psychological effects, right? When is a placebo needed? Involves humans, human subjects. And when do you not need a placebo? Yeah, animal testing, that's right. Okay, so I might give you a few different scenarios and I ask you which of the following situation requires or does not require the use of a placebo. Okay. All right, this was a quiz one question, right? What is a valid hypothesis? It must be what and what? That's right. Yeah. Testable and falsifiable. That's right. Now, by the way, like I just want to give you a fair warning, okay? Sometimes, like I know you guys don't read the questions too carefully, all right? Sometimes you read the multiple choice question and then A, B, C, D, E. You read A and you're like, oh, that's the answer, and then you move on without reading the rest of it. Please read all the options on the test because sometimes it will be like A, whatever, 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 and then E would be like both A and C. Okay? So like you might have to choose a later one. Do you understand? Yeah. Just telling you. Okay, write a hypothesis. So for example, if we think about the beetroot experiment, it would be an increase in temperature will increase membrane permeability. Okay. It's very straightforward. You know, if you took biology in high school, they might make you write it with a if and a then. Doesn't matter. Okay. For us, you just have to write a simple statement that includes your prediction of the relationship between two variables. All right, you remember this graph, right? Now, I know, I just want to reiterate, everything on this package will show up on the test, right? So if you see this graph on this sheet, that means you must understand it, okay? So over here, looking at the result, we have two graphs. The graph on the left here shows the control group, and then we have the treatment group. And you can see both groups experience a reduction in the jet lag symptoms. Based on this result, would you say the drug is effective in reducing jet lag symptoms? Effective or not effective? Yeah, this is not effective, right? Okay, so I'm going to type this one now. Okay, not effective because both placebo and treatment groups experience a reduction in symptoms, meaning the drug is not doing anything special than the placebo. In plain English, what this means is if taking a candy, you eat a candy compared to eating a drug, both are able to make you sleep better. Why would you eat the medicine or would you take the medicine instead of taking the candy, right? Understand? Because they are both as effective, right? Meaning the drug is not effective. The drug is not doing anything that a candy couldn't do. Not effective. All right. The graph on the right It's effective, right? Because, so effective, only treatment group experience drastic reduction in symptoms compared to placebo. Right? The candy is not supposed to do anything. The sham treatment is not supposed to have any effects, and it doesn't. That's why people who took the candy, I mean, they don't know they take the candy. That's the whole point of placebo. It's a double-blind experiment, right? So they have a lot of jet lag symptoms, and only the medication is able to reduce the symptoms drastically. Bottom line, if I give you these graphs on the test, you have to know whether, like, how to interpret them, right? Any questions? Like if, okay, one more time, like if you can't sleep, that's basically what this is, right? Okay. Taking the placebo, a lot of people still can't sleep, basically. Right. But taking the drug, less people will have the problem. So that's why the drug is working, right? Over here, people who take the drug or if they just take the candy, they both report improvements. So it's not really the drugs that's helping them. It's just, maybe more of a psychological effect, right? Make sense? Okay, that's that. And that is all for lecture 1. All right? Don't study more. If it's here, that's all you need to know. Okay? I'm serious. I know you guys have a lot going on this week. Any test tomorrow, by the way? Tomorrow? Okay, at least you have. I know. Yeah. I got the news. Yeah. As soon as because Michu sit next to me in the office. As soon as I saw her, she's like, "Oh, chemistry test is on the same day as your bio test." I was like, "Yeah, I know." No, I know. Because of the snowstorm. Yeah, blame the weather. It's okay. You guys will be fine. You guys will be fine. All right, this table, very important. Okay, elements for carbohydrates. Right? When I say elements, I'm talking about the individual stuff on the periodic table, right? Because when people get confused between monomers and elements, elements are just the carbon, hydrogen, oxygen, and whatnot. Okay, so for all of them, it's CH and O. Right? Some of them have more, but if you forget on a test and you just want to get like 0.5 or something, just put CHO for all of them. Okay. You can remember CHO, right? Okay. CHO. That's right. Then maybe you'll get 0.5. And if you're lucky, you might even get 4 marks if it's one of the carbohydrates. Okay. Now for proteins, though, in addition to CH and O, we also have what? Nitrogen. That's good. And traces of sulfur. Lipids is just CHO again, but that's right, traces of phosphorus, like the phospholipids that we talk about. The name phospholipid tells you it has phosphorus. Yes.
You have to write traces of, or can you just write the?
Yeah, it's okay. Just put, yeah. If you remember traces of, I'll be extra happy. If not, I'll just be like, if you see me marking your paper and I go okay, ... That's me saying, yeah, it's okay. It's like it's not what I'm looking for, but it's okay. I'll give it to you. Okay. You see a lot of these on your paper. That means you can improve your answer a little bit. Okay. Anyways, nucleic acid, CHO, and then 2 more. Yep, NAP.
Our math teacher, he says hi.
Oh, great.
And he says that he aspires to be you as a teacher.
He what?
He aspires to be you as a
Oh,
teacher.
he's too kind. I aspire to be him. Yeah. Okay, so monomers for carbohydrates. That would be monosaccharide. And by now you should know the different kinds of monosaccharides, right? So glucose, that's right. Now that's a good question. Hold on. Again, I'm just trying to be transparent with you guys. You are not gonna have to fill in a chart like this on the test, but there will be multiple choice questions that will ask you the information on this chart, right? So like, do you have to list the monosaccharides? Probably not, but there are a couple ways I can ask you, right? Which of the following is the monomers for carbohydrates? Then you know to pick fructose, right? As opposed to amino acid or whatever, right? Monomers for proteins. What is it? Not that one. Someone else? Amino acid. Yes, yes. For protein is amino acid. Yeah, amino acid. Now it was weird because on the quiz, people instead of writing amino acid, some of you drew the amino acid, which I thought would be harder. But yeah, always answer with words, right? Unless I ask you to draw. And there will be some drawing on the test, right? So bring some colors to help with the drawing. Nothing fancy, just like maybe red and blue. In addition to your pencil would be okay. That's fine. That's fine. I mean, like, it just makes it easier for me, right? Lipids would be various components, right? So we have things like glycerol, fatty acids. We have like carbon rings for the steroids. And we also have phosphate heads, right? Phosphate head. Yeah. Yeah, sorry. And the monomer for nucleotides are, sorry, I just gave you the answer. Yeah, the answer for the monomers for nucleic acid is nucleotides. Yeah. And just in case, nucleotides has phosphate plus sugar. It's either ribose sugar or deoxyribose sugar, and a nitrogenous base. Yeah. ACTG, right? And then ACUG for RNA. And sometimes if you are stuck on a written questions or like you forgot, how to spell something, just flip through the multiple choice sections, okay? Like you might be able to find some of the words, right? Okay. Like some of you forgot like the ACTG stuff, but then there was a question about the ACTG in the multiple choice, right? So, use that to your advantage. All right, biological function. So I'll do these two things at the same time. For example, we have starch. Starch is the storage form of energy in plants. What about us? What do we store energy as? Glycogen, that's good. Storage form of energy in animals. There is also things like cellulose. which is structural support for plants. And by now you should know cellulose are the key component of what portion of the cell, of the plant cell? Cellulose makes up which portion of the plant cell? More specifically for the cell itself, cell wall, that's right, okay? So e.g. cell wall. And then chitin. Chitin is exoskeleton, right? Of crabs, yeah. Crabs, shrimps, mussels.
Maybe insects.
Insects, yeah, as well. Very good. If you don't have enough time and you don't know where to start, just copy these things over and over again, okay? Don't even make your own notes. Just copy and try to memorize as much as you can. You should be okay. All right, proteins. Again, we use proteins for structures, right? Structures, e.g. like muscle and things like hair or nails. We also use proteins as hormones. That's right. So an example of hormones would be insulin. That's the only one we learned. Which is an enzyme, yeah, enzyme. An example of enzyme would be like lactase, right? You just change the last few letters to ASE. So lactose is broken down by lactase. Don't forget, membrane proteins are very useful as well. And then there are 6 different functions that we talked about, right? You need to memorize at least 4 of these things, okay? You need to. So for example, cell recognition, yeah, transport. That's right. Enzymatic activity. Oh, good. Are you reading it off? Oh, you are. Okay. I was like, oh, good job studying. But yeah, attachment. Yeah. Oh, good. Attachment to cytoskeleton. Intercellular junction. Okay, and then there's one more, which is, that's right, cell signal. You know, the Halloween story. Okay, cell signaling. All right, so like I said, remember these ones. 4 of them at least. Okay. Remember everything I'm writing, but again, if you're short on time and you don't have enough memory load, then this is, first thing you should get into your head. All right. I cannot be more transparent than how transparent I am being right now, okay? I hope you're picking up the not so subtle hint. For the lipids, lipids can form like a waxy barrier. So barriers to water. Lipids can also be hormones. So like testosterone, estrogen, progesterone, if you want. And lipids can also be used for storage, form of energy, We didn't talk about that one, I don't think, but it does, yeah. Okay, storage of energy. That's pretty much it. And oh, and of course, we have the phospholipids, which is a major component of cell membrane. Okay. These are, by the way, called like steroid hormones, right? If I show you a picture of steroid, you would recognize it with the 4 rings, right? Okay, nucleotides, we've only learned 2 types. We have DNA, which is instruction to make proteins. RNA are basically copies of DNA. Okay. There are different types of RNA responsible for different things, but we've only mentioned them by name. You don't have to know the exact function. And that is 1 page down. Let's keep going. Give me a second, I'll put those things back on. Okay, well, what is this first one here? Amino acid group, nucleotide. Glucose, carbohydrates, yeah, glucose probably better. I mean, we don't know for sure if it's cholesterol, but it's definitely steroid. Yeah. Okay, good.
What did you say the third one should be?
Yeah, I'll write down.
Instead of
Glucose would be okay. Nucleotide. Okay, glucose or monosaccharide, that's fine. Obviously, this is for protein. Nucleotides are for nucleic acid. This is for carbohydrates, and steroid is a type of lipid, right? Yeah. All right, compare similarity and differences between saturated and unsaturated. And again, this is one of the things you should definitely prioritize if you have limited time to study. Okay, prioritize. All right, here we go. Okay, not fatty acid, fat, okay? Fat. So elements, CHO for both of them. Components, please be careful. 1 glycerol plus what? 3 saturated. Yeah, 3 saturated fatty acids. For unsaturated fat. Plus at least 1 Unsaturated. You can have more than one, but you need to have at least one. Yeah. If the question gives you line, then explain in sentences. If I give you a table, then you fill in the table. It's gonna be a table, okay? I'll tell you. It's gonna be a table. Stay at room temperature. And for saturated fat, is it solid or liquid? Yeah, okay, mostly solid. And then this one is mostly liquid. Double bond, yes or no for saturated fat? Huh? Double bond?
Yeah.
Yeah. No, maybe. Yes or no? No, yeah. No, okay. Absent. Yeah, they don't have any. Yeah, trust me. Unsaturated, present. Number of hydrogen atoms, max. Okay, this one not max. Has room for more. Example. Give me one example of saturated fat. Butter. Okay, please don't make up complicated examples. Just use the one I give you. Okay, butter. How about what kind? Sure. Okay. Name your favorite oil. Just not coconut, okay? Coconut is saturated. Everyone still okay? Any questions? All right.
Sorry, what is that,
Oh, max number of hydrogen atoms. So for saturated fat, it has no more room for hydrogen. All the carbons are filled with hydrogen. Okay, whereas this one, because of the double bond, it still have room for more. All right. This question, if you supply radioactive nitrogen atoms, if you don't know what that means, the bracket says something that can be traced and detected. If you supply these radioactive nitrogen atoms to a cell and let it grow, what molecules will you expect to find the radioactive labels in? Basically, this is just a complicated way of asking you, where do you find nitrogen atoms in macromolecules? So if we look at our table up here, which one has nitrogen? Proteins and nucleic acid, right? Okay, so that's what will happen. If you trace all the nitrogens in the cell, they will all show up in either the proteins and nucleic acid. Okay, DNA or RNA doesn't really matter.
So sorry, the wording that you just used to make that whole sentence simpler, what did you just say?
Which one? This one?
Where do you find nitrogen atom? Yeah. just
Oh,
the
yeah.
wording.
Sure. Where do you find nitrogen atoms in macromolecules? Okay. What is the monomer for DNA? Again, we said it's nucleotides. So we have the phosphate plus deoxyribose and ACT or G. This is the nitrogen base, right? A nitrogenous base or a nitrogen base, doesn't matter. That's okay. Yeah, it's fine, it's fine. Okay, how are nitrogenous base classified? That's right. So purines are A and G. You should know that by now. How many rings do they have? 2, yeah. Pyrimidines, they are T or U and C. Yeah, see you later. 1 ring. Now, as for the base pairing rule, what does A pair with? And then C with. That's what's holding the nitrogen base together? Like you have a strand here. That's right. Okay, so like this is one strand, this is the other strand. So what's holding them together would be these hydrogen bonds. And remember, there are 3 between C's and G's, and only 2 between A and T's, right?
Where it says like the pyrimidines, then it says T, what's after that?
T or U, uracil, uracil. There we go. What is the significance of base pairing rule? So we have uniform width across the DNA, right? It's always 3 rings across. And also, the strands are complementary. If you know one side, you will be able to figure out the missing side. You will see in the second half of the course that is very important when it comes to copying our DNA. The strands are complementary. No, higher up. Uniform width across the DNA. Okay, what is the difference between denaturation and dehydration synthesis? Denaturation is when proteins are proteins unfold due to temperature. What else can unfold a protein? Yeah. Yeah, ion concentration. Type of solution. like whether it's hydrophobic or hydrophilic. It's going to cause the proteins to unravel. Example, egg white, right? Cooking egg white. When changed from transparent to white, that's basically protein denaturing. Okay? There's no going back. Dehydration synthesis is not really related to denaturation. Dehydration synthesis is when you have monomers and it becomes polymers, right? When you take individual building blocks and you join them together, that's dehydration synthesis or condensation reaction. Okay.
For the dehydration, if you ask, you have to explain the whole thing, like this, well, the other side is.
It's hydrolysis, right? Yeah, yes. Yeah, I mean, just know the relationship would be nice. Hey, can I pass the attendance for you guys to do it? It's quicker if you do it than me. Hey, I think that's it. That's it for lecture 2. 3 more to go. Any question? You guys look exhausted. You'll be okay. Okay. So, no diagrams, like the quiz. I won't give you a cell to label, but I might give you an organelle in a multiple choice and ask you questions about it. So I might show you a mitochondria, and then I say what reaction happens in the organelle shown. And you have to know it's cellular respiration, right? Okay. I will also have a question where I give you the functions, and you have to tell me what organelle does that. Okay? You hear that? I'll give you the functions, and you tell me which organelle. And I will even give you a word bank to choose from this time. Okay?
Yay.
Yeah. So, you know, people always miss the word bank, so I make the word bank font 24. Hopefully you will see it. Okay, so I'm just gonna give you one-liner for these things here, and hopefully it will make it easier for you. Okay. Now, since you guys having trouble reading what I'm writing, maybe I'll just type this part. Okay, a ribosome in one simple, a few words. What do ribosomes do? Oh, good. I feel better. Protein synthesis. Okay. Similarly, for mitochondria, 2 words, 3 words. Energy production, very good. Site of cellular respiration. Yeah, they receive vesicles and they sort, modify, and repackage proteins arriving in vesicles. I'm going to make these a little bit bigger for you. Oh, that's too big. Sorry. Rough ER works with ribosome to produce and package what? 13th. Yeah, into vesicles. Very good. Yeah, lipid synthesis and breakdown. It's probably good enough. And of course, they do detoxification as well. Maybe put that in as well. Nucleolus. Yes, very good. Produces ribosomes. The nucleolus is inside the nucleus, right? Nucleus basically that's where you keep your DNA. Lysosomes, they contain enzymes, so they break down and recycle macromolecules or old organelles. And also kill the cell if it becomes too old or if it's not functioning properly. What reaction takes place in the chloroplast? That's right. The cell wall, which you know is made up of cellulose, it protects the plant cells, prevents bursting. What kind of solution do we put a plant cell in so that it swells up? Hypotonic. Very good. We'll review those in a bit. Central vacuole stores water, right? And nutrients. There you go. I might have missed one or two just in case, okay? Cilia, it's basically helps with movement, right? Covers entire cell body. What's the other one? That's right. Helps with movement. Usually just one or two per cell. Okay, that's all. I think I have a matching question in the self quiz that allows you to practice with this. Which one, Ruth? Nothing? Okay. This is more important than the self-quiz, okay? This is more important than the self-quiz. All right, central dogma of biology, central theme. So information stored in DNA are copied in the form of messenger RNA. And then from that, it is going to be decoded and used to build proteins. What do we call this process from DNA to mRNA? Transcription. And that takes place in the nucleus. What about from mRNA to protein? What is that? That is translation. And that happens in cytoplasm. Okay? Or rough ER. Yeah, I can take it back. Thank you. Next, describe the structure of a mitochondria and state the function of each part. I mean, we probably do this again later, but maybe very briefly. That's the outer membrane, right? And what do we call this thing here? Starts with a C? Yeah. Cristae. And that's where the ETC happens, right? Over here is the matrix, which is where pyruvate oxidation. That's right. And Krebs cycle. This is the intermembrane space, which allows H plus concentration to build up. Okay. Pyruvate oxidation here plus Krebs cycle, KREBS cycle. Intermembrane space, remember, is for the buildup of the H plus. The whole point is to create a lot of H plus here so that they want to go back in to the matrix. Right? And in doing so, they pass through the turbine and it generates ATP. This kind of ATP production is called oxidative phosphorylation. I'll write down for you later. Pyruvate oxidation and Krebs cycle. Stage 2 and stage 3. This one? Sorry. Are we okay? That is it for lecture 3. Number 4. We talked about this in class, I think. So if you want to increase membrane fluidity, in other words, it makes it more fluid, then do you want higher or lower cholesterol? Higher, okay, higher. Remember, the cholesterol keeps the phospholipids apart. They prevent them from binding together. The more apart the phospholipids are, the more leaky it's going to be. Okay. If you have trouble understanding this, like think about building fences, right? To keep, I don't know, chickens in the pans or something, okay? So these are the fence. If the fence are too far apart, then the chicken's just gonna, escape in between, right? Okay? So you want these things to be really close together, more solid, so there are no gaps. Now, what are the cholesterol? Cholesterol are like things that are standing in between, and it prevents the fence from binding together. If the fence don't come together, then there are gaps and things can leak, right? And that's what this is about. Okay. If you want to increase fluidity, if you want things to leak out, in other words, you want more cholesterol. From your lab, if you want things to leak out, do you want higher temperature or lower temperature? High. Who said lower? Higher, okay? Higher temperature. And you need to heat it up. The more you heat up, the more color comes out, right? Okay. Unsaturated fatty acids, more or less? Okay, this one is also higher. Okay, remember unsaturated fatty acid has the kink, right? So with the kink, you cannot pack them closely. There will be a gap and they will leave, things will leak out in between them. Okay, lower like that. If you need to refresh the reason for this, go back to lecture 4. At the beginning, there is a slide. After we drew the whole cell membrane, I had a slide to talk about the plants, okay, in the cold environment, hot environment. Go to that one to revisit it. Okay, so everything is opposite for the other one. Number 2, list 6 functions of membrane proteins. We did already. And then there's the chart. We drew the chart last Tuesday, remember? I did it on the board, right? That's the chart you need to review. Yeah, I'm not gonna go through it again. I posted for you already, I believe. It's this one, right? This chart right here. Okay, so I can ask you different questions from this. The most straightforward questions would be, which of the following is an example of active transport or passive transport? So you have to know what belongs to what. Okay. I can ask you which of the following requires ATP. That's just another way of asking you which one is active transport, right? Because only active transport requires ATP, right? I can ask you which of the following involves a membrane protein. You should write these down. Membrane proteins would be like, channel protein, carrier protein. What else requires a membrane protein? The protein pump, right? So just
Wait, why?
requires the help of a membrane protein. So things like diffusion or osmosis, they don't require proteins, right? But facilitated diffusion requires protein. Protein pump requires protein. Okay. Familiarize yourself with these terms. And, if you need help, just email me. Okay, and that's next page.
You have to put it, be able to put it in the right font for the.
No, there won't be a chart like this on the test, but the test will have questions related to this chart. Okay, so if I ask you, which of the following is an example of endocytosis? Okay, so look at the chart. Okay, go find out for me. What are examples of endocytosis? Look at your class notes and tell me. Huh? What is it? So yeah, so pinocytosis is the one example. What else? Phagocytosis, and the last one, receptor-mediated endocytosis, right? Okay, should be at the far right of the chart. So that's what I mean. You need to know the terms, like what's the relationship between them. Okay. All right. What? Okay. So here is the next one. What question is that? Is it 4? Question 4. What are some factors that affects the rate of diffusion? So we talked about 4, I believe, in class. Size. The bigger it is, the slower it will be. And what else besides size? The steepness, yeah. Steepness of concentration gradient. Charge. And there's one more. That's right. If it's hot, it tends to go faster. Right? We did a note on this. Do you remember which way water will move? The water will move to the right side, right? If the dot's not going to move because the membrane is selectively permeable. And so water, which will always be attracted to a location with higher salt concentration. And this is called osmosis. Osmosis. So water is going to go to the right. And so the water level will come down here. Water level will go up. And then this gets diluted. And on the left side, because you're losing water, what happens to the concentration? What's the opposite of getting diluted? Yeah. No, that's it. You have it, no? Concentrated, yeah. It's getting concentrated. Okay, so this one's gonna get diluted, decrease in concentration. This one is gonna increase in concentration. When will the water stop moving? Water is gonna move to the right, but when is it gonna stop? When there's what? Not necessarily. Okay. Water's gonna move there.
Well, concentration.
That's right, when concentrations are the same. And once they're balanced out, once you've diluted enough and concentrated enough, and they're the same, then there's no more forces drawing water one way or the other, right? Okay, let's go through the three cases for the tonicity. Hypertonic. Hypertonic. Solution. The solute concentration, solutes are the dots, right? The salt. The solute concentration in the solution is greater than. That's what hyper means. Greater than the solute concentration inside the cell. Rate of water entering the cell is, that's right, less than. Let's draw this out. Yeah, so there's a before and there's after. So this is the animal cell. This is the solution. And then I'm gonna put more dots on the outside, right? You should put like 1 or 2 on the inside as well. But do make a point that there are more on the outside. And so water will be leaving. And water will be coming in. Do make a point to show me there are more leaving than coming in, right? Yeah, higher than is fine. Higher than, greater than.
And we're gonna actually have to like draw them in.
Likely. After, what's going to happen to the cell after? It's going to shrivel up. It's going to shrink up, right? Okay, that's it. We have a name for this, don't we? That's right. Is the animal cell doing okay in this case? Not the best, right? But not going to die either. Okay, I'm just going to quickly do the other two. We actually took a note on this, right? Like this one, right? So this is lecture 4. Go look at it again if you need to. It's what is it? This is on February 4th. Okay, if you need the note. February 4th. Okay. Excuse me. Thank you. Isotonic. The solute concentration in the solution is the, yeah, it's equal to. And the rate of water entering the cell is also equal to. Okay. So how many arrows going in? Yeah, 2. 2 arrows going in and then 2 coming out. What's going to happen to the cell? Nothing is right. And after is the same. This is too easy. I probably won't give you isotonic, right? Yeah. And don't memorize greater than and less than, okay? Like it really depends on how it's being phrased, right? I am not always consistent myself. Like here it says solute in the solution and then solute in the cell. Like it might be the other way around on the text, right? You really have to read it. I cannot promise you it's in the same order. Okay, hypo, the solute concentration in the solution is less than. And then this is greater than, right? greater than. And it should make sense to you. Don't just memorize them. Okay, and this time, more dots where? Inside, right? Yeah, inside. Yeah. How many arrows going in? 3 arrows going in. Yeah. That's why colors would be nice. And then 1 coming out. Before. And what's going to happen to our cell? It's going to burst. Okay, so you can do burst like this. Okay. Lysis. That's the fancy word for burst. Okay. Opposite of lysis is plasmolysis. Plasmolysis. Okay. Plasmo and then lysis. Oh, we're almost there. I know. That's it for lecture 4, huh? Lecture 5, which was like very complicated, lots of things, but just know these 6 answers to these 6 questions, then you should be all right. Okay. Oh, this is a 2-part question. Oops. Okay, write a balanced chemical equation. I told you you must know this for the test. Oh, yeah, sure. Go ahead. Yeah.
C6, H12, O6
Mm-hmm.
plus O2,
How many?
6
Yeah.
equals to 6 CO2
Mm-hmm.
plus H2O, I think also 6.
That's right. Very good. How many ATP? 36. That's right.
6 ADP.
Yeah, 36. Very good. But that's not it, because the question says clearly indicate a reactant being reduced and oxidized. So this guy is going to be, which one is, what's going to happen to glucose? Oxidize, correct? You're going to rip electrons from the glucose, right? And those are very energetic electrons, which we will ultimately use them to pump the hydrogen ion across the inner membrane. These ones, reduced. That's all I am asking you to remember for the equation. All right? You can do it. What is the role of NADH and FADH2? They basically transport, they're the taxis, right? Transport electrons to ETC. Okay. They capture the electrons from stage 1, stage 2, and stage 3, and then they take them to the ETC. Assume a mitochondria contains 50 NADH and 19 FADH2. If all the energy carriers were used to make ATP, what is the maximum number of ATP that could be generated from oxidative phosphorylation? 250? Yeah, okay. I don't know, I didn't do it. Oxidative phosphorylation is the pumping the H plus over. It's the stuff I explained to you last Thursday online, okay? If you missed that lesson, please watch it. It's important. that you understand it. Okay, so let's do this. This is 50 times 3. Let's do 150. 19 times what? 2. Okay, the 3 and the 2 are fixed, okay? It's always times 2 for NADH, always times 2 for FADH2, and that's 38, yeah. And if you add it, is what? 188, okay? I don't think you need a calculator, but if it makes you feel better, that's fine. You can bring one. Okay, nothing weird, okay? Don't tuck papers in there. Don't use pencil to write on the black cover. I know all the tricks. Anyways, next one. What is the maximum number of ATP that can be produced with 9 turns of Krebs cycle? Okay, this one also very important. I already started for you in the lecture notes, but I'm gonna start again. And I'm gonna ask you to do it. And I'll give you, what, 2 minutes maybe, and then I'll take it up with you.
81.
How many?
81.
81?
Actually, 9 times, wait, 9 times 12 is 108.
108, okay. Anyone else? Also 108? Yeah, yeah. You gotta show your work though, okay? All right. On the test, if it says show your work, and if you only say 108, I can only give you 1 mark. And don't copy the person next to you. They might not have 9 turns. They might have 12 turns. Anyone else have something different? Basically, this is what you need to remember, okay? This is 1 turn, right? You don't have to draw this or anything, but you just have to remember the key numbers from it. Oh boy, this is 1 turn of the Krebs cycle. Okay, so 1 turn of Krebs cycle produces 1 ATP. This is by substrate level phosphorylation. It's the direct production of ATP, right? With the help of the enzyme, right? There will be 3 NADH and 1 FADH2. These energy carrier go to ETC for oxidative phosphorylation. So this is 1 ATP. This one is times 3. And this one is times 2. 3 times 3 is 9. 2 times 1 is 2. So if you add these up, that's 12 ATP per turn, right? And then the question asks you for 9 turns.
Sorry,
Mhm.
what did you write? Go to ETC
For oxidative phosphorylation. All right here. Oh, I don't have it, but oxidative phosphorylation. Here, I'll spell it out. It's the whole pumping H plus thing. That's what it's called, right?
Oh,
Yeah, oxidative. So 9 turns times 12 is in D108. Okay.
Always 12 ATP per 1
Per cycle, that's right. But then you have to tell me where they came from. Right, Kathy, which word? This one? Yeah, sorry. PHOS. O-R-Y, and then alation. Yeah, L-A-T-I-O. So remember, there are two ways of creating ATP. Substrate level phosphorylation, which is only 10% of all ATPs. And then there is the oxidative phosphorylation, which makes up 90% of the ATPs. Which step of cellular respiration generate the greatest number of ATPs? It would actually be ETC, right? Because with all the energy carriers coming in. Okay. Now I show you this on Friday already, but let me just go through it very quickly for you one more time. You don't have to draw this. And you just have to remember the whole point of this situation is for the energy carrier to be coming in and deposit the electrons. Okay, so they're going to put the electron over here or at a lower point. And as the electron are being passed down the ETC, we will pump H plus from the matrix into the intermembrane space. Okay? And I told you last Thursday, this is like building up a dam with all the water. They want to come back in, right? But the crystal is blocking it. So the only way to come back in is through the turbine, like the ATP. So when they come in through the turbine, and when these things goes through the ATPase, it generates ATP. Okay. What is the purpose of the final electron acceptor? Can you tell me what the final electron acceptor is? Not water. That's right. But what is it? Like what molecule is the final electron acceptor? Oxygen. Yeah, this is O2. And the purpose of O2, and I wrote it down for you, is to remove electron from ETC to allow continuous electron flow. So oxygen must come and catch the electron at the bottom and take it away to form water before the next set of electrons can slide down. If we don't have oxygen, then the electron cannot flow. No electron flow, no H plus, no ATP. We die.
Can you write out number 5 in words?
Can I write down number 5? Describe what happens. Yeah, I think I actually did that. Let me just copy it from last class here. There. As electrons are passed down the ETC, its energy is used to pump H plus from there. I'll copy it over. So that's all. You just again study this green sheet and also study the questions we do in class, right? Those poll questions that I asked you to do 1, 2, 3, 4. Right? Some of those will be on the test. Let me just show you where the answers are in case you don't know. I'll post this as well. So, if you missed something, you can find it. It's over here, guys. Under test 1, there is the answers. Okay? There are the answers. Because no, that's basically this review video, right? Yeah. Okay. Good luck, guys. Don't miss the quiz, okay? The test.